Scientists assembled this cell from components that were never alive. Then it fed, grew, copied its genome, and divided. That sequence sounds less like chemistry and more like a birth announcement. The system is called SpudCell. In a preprint posted in July 2026, researchers at the University of Minnesota report a chemically defined synthetic cell capable of completing a laboratory cell cycle across multiple rounds. But here is the twist. SpudCell still cannot live independently. It cannot build some of its most important machinery. It must be fed packages containing material borrowed from living cells. And during the main 5-generation experiment, the researchers physically helped it divide. So did scientists create life, or build an extremely convincing machine at the border of life? This is not just a word game. The answer changes what we think a cell is, how little machinery biology truly needs, and how close engineers may be to constructing living systems from basic components. There are 2 basic ways to search for the smallest possible cell. One is top-down. Start with a living bacterium and remove genes until further deletion kills it. This reveals what an already living system cannot do without. The other is bottom-up. Begin with purified molecules, DNA, proteins, lipids, salts, and ask whether the right combination can cross from organized chemistry into something lifelike. Top-down is like stripping a running car to its essential parts. Bottom-up is receiving a garage full of metal, rubber, and wiring, then discovering whether any arrangement can start the engine. Biology, inconveniently, does not include the assembly manual. SpudCell belongs to bottom-up engineering. Its membrane is a lipid vesicle, a hollow sphere made from fatty molecules like those in natural cell membranes. Inside, it carries a genome of roughly 90,000 DNA letters. That genome is split among several DNA molecules. The vesicle also contains a defined protein-making system built from purified biological parts. Every starting component and its concentration can, in principle, be listed. That makes the system useful even if the philosophical question, alive or not, remains unresolved. SpudCell's cleverest feature is how it eats. It does not digest food or manufacture everything from raw nutrients. Instead, researchers surround it with smaller packages called feeder liposomes. These carry fresh membrane lipids, enzymes, ribosomes, and small molecules. SpudCell's DNA encodes a modified membrane protein derived from alpha-hemolysine. The cell's internal machinery reads that DNA and produces the protein. A tag on the protein can connect with matching molecules on the feeder package. The 2 membranes fuse, and the feeder's contents enter the synthetic cell. The important step is not merely that 2 bubbles merge. The genome controls production of the protein that enables the merger. More expression can mean more feeding can mean more membrane, more resources, and more growth. For the first time, a useful chain begins to appear. DNA influences a trait. The trait changes access to resources. Access changes reproductive success. That chain is one of the foundations of natural selection, but there's a large nutritional label on this meal. The feeder packages contain complex machinery that originally came from living biology. SpudCell is not building all of its own parts from simple chemicals. Life is still helping from backstage. Before cell divides, it must copy its genetic information. SpudCell uses a DNA copying enzyme called Phi29 polymerase. That enzyme originally comes from a virus that infects bacteria. The researchers measured newly synthesized DNA after feeding cycles, along with RNA and protein production. In the main experiment, each cycle lasted about 12 hours. The synthetic cells received feeder liposomes, copied DNA, grew, and were then pushed through a membrane filter to split them mechanically. This was repeated for 5 rounds. That result matters, but 5 generations can sound more autonomous than the experiment actually was. For these runs, division was assisted by laboratory extrusion. The researchers chose this method because it produced daughter compartments more reliably while they tested feeding, replication, and selection. There was another problem. The genome was divided among several DNA molecules, and SpudCell had no chromosome segregation machinery to guarantee that each daughter received a complete set. After 5 cycles, approximately 30% of analyzed cells contained the full multipart genome. The others were not tiny independent organisms ready to colonize the laboratory. Some had lost essential instructions in the shuffle. Copying DNA is impressive. Delivering every required copy to the correct daughter is a different engineering problem, and natural cells have spent billions of years becoming annoyingly good at it. The team also tested a division mechanism controlled by genetic information rather than a filter. Natural cells use coordinated molecular structures, including cytoskeletal machinery, to organize division. Rebuilding that system from purified components is brutally difficult. SpudCell takes a shortcut. It produces proteins that accumulate at the membrane. When enough crowd together, they can deform the surface, create mechanical stress, and help the membrane pinch apart. In a separate experiment, the researchers connected expression of these membrane-associated components to division. Cells with the relevant genetic program produced more daughter compartments than controls. This is the most visually lifelike result. A genome is read, proteins appear at the membrane, and the compartment changes shape until one becomes more than one. But the yield was lower than mechanical extrusion, and outside helper molecules were still involved. The system did not demonstrate an indefinitely self-sustaining lineage, driven only by materials it could make for itself. It showed that genetically encoded division can work in a chemically defined synthetic cell. That is a major step. It is not the finish line. Then the researchers created competition. They compared synthetic cells carrying different versions of a promoter, the DNA control sequence regulating production of the feeding protein. One version drove stronger expression. Those cells fused with feeder packages more efficiently, grew more, and produced more daughter compartments. Across repeated cycles, the stronger feeding variant increased its share of the population. When resources were limited, its advantage became more important. That is selection. A genetic difference produces a physical trait, and the trait changes reproductive success. But it was not open-ended Darwinian evolution. The advantageous change did not arise spontaneously. The researchers designed and introduced it. The preprint itself identifies spontaneous mutation and continued evolution as a future step. Evolution was invited into the laboratory, but it arrived with a reservation. Even with that limitation, the experiment matters. Previous artificial systems could copy molecules or divide compartments. SpudCell links more of the chain together. Genome expression, resource acquisition, growth, replication, division, competition, and population change. The achievement is not any single trick. It is getting the tricks to affect one another. So why not simply call SpudCell alive? Because life is more than performing several biological verbs once under carefully prepared conditions. SpudCell has extremely limited metabolism. It cannot manufacture ribosomes, the molecular factories required to make proteins. Its feeder liposomes must deliver ribosomes, enzymes, nutrients, and membrane material. Its genome inheritance is unreliable. The strongest multi-generation result used mechanical division. The genetically encoded division experiment still depended on external components, and the work is currently a preprint. It has not yet completed peer review. The measurements may be valuable while interpretations, methods, and claims are still challenged or revised. There is also no universally accepted checklist that turns chemistry into life at one precise line. Metabolism, reproduction, heredity, evolution, and autonomy all matter. But familiar examples refuse to cooperate. Viruses evolve but depend on host cells. Sterile animals are alive but cannot reproduce. A flame consumes fuel and grows, yet we do not give it a genome or a family tree. SpudCell does not settle the definition. It makes the definition uncomfortable. Scientists have not created a fully autonomous organism from simple chemicals. What they report is narrower and, in some ways, more useful. A defined synthetic compartment in which genetic information can control feeding, growth, genome replication, division, and selection like population change. Because the parts are known, engineers can alter the system deliberately. Future synthetic cells might become tiny chemical factories, programmable sensors, or controlled research platforms. Those applications remain possibilities, not results demonstrated by this preprint. The next milestones are difficult. Build ribosomes from genetic instructions, create a more complete metabolism, distribute the genome reliably, improve autonomous division, and allow useful mutations to arise rather than be inserted by researchers. If those systems eventually sustain themselves, the argument over whether they are alive may stop being philosophical and become practical. SpudCell is not life manufactured from nothing. It is life's machinery reorganized into a new system, one that can perform a surprising portion of the cycle while still depending on biology outside its membrane. That dependence is the boundary. For now,